Well, thank you for attending today. We have an alumnus with us, Dr. Elda Rueda. She's both an adjunct at University of Houston downtown and works, her primary work is at the Baylor College of Medicine. So we're very excited to have her. We have had Dr. Rueda as a keynote for the Student Research Conference years ago. Excellent student. I happened to be able to invite her to help us with the Board of Trustee award that the Scholars Academy was able to garner out of the four universities of Houston system. So we were very thankful for that. Today, she'll introduce herself and introduce her topic. And without further ado, Dr. Rueda.
Sorry. No, just one moment, please. Okay. Okay. Thank you very much, Dr. Parker, for the introduction. And thank you very much for inviting me to give this presentation. It is always an honor for me to be part of Scholars Academy and UHD events. And I love the fact that I keep this relationship with UHD after I graduated from UHD in 2011. So I realized that for this presentation, one of the most important central interesting parts is the research, and so in the science. So I'm going to start by presenting my research project, and as I show you the data, I will tell you the story and how I became interested in my research and how I move a step from one step to the next. So I studied the neural retina. And I first learned about retina as a tissue model for neuronal biology, to study neuronal biology, when I was a senior undergraduate student at UHD. And I'm still working on research, on retinal research after 10 years. So that is not an example of lack of imagination, but it's actually an example of perseverance. and people who do research understand that part. And so retina is an extension of the central nervous system. And as part of the CNS, the retina is susceptible to several to neuronal loss due to either congenital diseases or degenerative diseases. And so the retina is a tissue that lies in the back of our eyes, and it is directly responsible for absorbing the light and sending those signals to the brain for visual perception. And so there is a number of common retinal degenerative diseases that result in irreversible blindness. And among them are diabetic retinopathy, glaucoma, and also age-related macular degeneration. And for years, the National Eye Institute had been collecting a lot of data. And what they have shown is that particular diseases are prevalent in particular ethnic groups. And so one example is diabetic retinopathy. It is very, very common in the Hispanic population. And so there are several attempts to repair the retina. And one of them is, for example, gene therapy. This is a very promising, and in fact, a few years ago, the first AAV or gene therapy was approved by the FDA. But this is only to repair retina for congenital diseases. There are more experimental strategies that are being developed, and one of them is cell replacement therapy, for example, which it's still very in the early stages. We have to make sure that when we transplant these cells, we're not creating tumors, for example, and that is very challenging to do. And we also have some pharmacological tools that we can use, but they only delay the loss of the neurons. And so that's what is common about any retinal degeneration, is that the loss of neurons. And none of these strategies so far are able to address this issue. And so the general question still is, how can we regenerate the tissue, and how can we produce the neurons and recover visual perception or a vision? So one of the audacious goals of the National Eye Institute and several researchers is to regenerate these neurons utilizing an endogenous source of cells, and that will become a little clearer in a minute. So basically utilizing a particular source that it is living within our retinas. And so to move forward with this goal, we have to know every single detail about the retina. And retina has been studied for 100 years, more than 100 years now. And I started learning about retina when I was an undergraduate student in the lab under the mentorship of Dr. Jerry Johnson. And I became fascinated with this research and with the tissue, because for the first time, I started to connect what I was learning in the classroom with something that was related to biomedical research. And so to me at the time, it sounded that I was going to be doing something important for medicine. And so it became very, very interesting and became a big part of my undergraduate life. So based on the literature, I started to learn that the retina is a very unique piece of tissue. And although it is part of the brain and it does function like brain cells, when we started to study the metabolic profile at the gene level, we found that this tissue looks very much like muscle cells, like liver. And we also saw that they were it was even expressing some genes that are normally expressed in highly proliferative cells like tumor cells. And so it became really, really interesting to me. So after that, I joined the Vision Science program at the College of Optometry at the UH main campus for my PhD training under the mentorship of Dr. Donald Fox and her student at the time, Dr. Shantay Shanay. And so I learned a lot more about the retina, how the tissue develops, how the tissue is affected, those cells are affected by particular neurotoxins, and how it is affected in degenerative diseases. And so to give you more details about this, the retina is composed of several neuronal types, and one of those are the photoreceptors. Those are the cells that are directly responsible to absorb the light and transduce that into a more conventional neuronal signal that is then processed by interneurons that are living within the retina, and they're passed along into these line of retinal ganglion cells, which are directly connected to the brain. And so these cells, I learned that they are affected by different toxins at different developmental time points. And one thing that we found is that, for example, lead toxicity during development affect primarily photoreceptors and interneurons. And so that was mostly the work led by Dr. Shantay Shanay when I was a student. She became my guide during several years of graduate school. And after that, we became good friends. We are still friends. And at the time after, as a result of reading the literature, going to seminars every day as a graduate student, and I started really engaging in the lab work, I became very interested in the glial cells of the retina. And so, as many other regions of the brain, this retina contains glial cells, and they're highly specialized. They're called the Mueller glial cells. Their cytoplasm surrounds every single neuron of the retina. They participate in recycling of neurotransmitters, and they allow the metabolic exchange between neurons and the blood-brain and the blood vessels. They maintain the osmolarity, so they're very, very essential. They play very essential roles in the tissue. Now, these cells in the zebrafish and in non-mammalian and few non-mammalian vertebrates have this remarkable ability to reprogram at the molecular level. And after damage and a loss of neurons, they become stem-like cells that they can undergo several rounds of cell division and at some point become neurons. And so they have this amazing regenerative capacity that the mammalian species, including human, do not have. However, because all mammalian species, including humans, have the same exact Mueller glial cells that play the same exact roles, potentially the cellular regeneration capacity is present in our retinas. But what it requires is that we identify those molecular targets that can induce regeneration. And so to start figuring out how and why these mammalian species cannot regenerate the tissue through these Mueller glial cells, for my postdoctoral training, I joined the lab of Dr. Ross Pochet, and with two of his collaborators, we identified the hippo pathway as one of the signaling pathways or mechanisms that block this Mueller glial cell capacity for proliferation. And so this pathway was first discovered in Drosophila several years ago. And later it became obvious that it was an evolutionary conserved pathway that during development regulates tissue size. So this is how it looks when the pathway is not turned on. And so that the pathway itself maintained that by balancing cell proliferation, cell apoptosis, cell renewal. And after many years of several years of research in several labs, it became evident that the hippo pathway play a key role in adult tissue regeneration. And that is because certain cofactors like YAP regulate a switch between a proliferative and non-proliferative state. So for the next 3 years, we dedicated the time to utilize and develop several pharmacological and molecular as well as genetic tools that allow us to isolate these Mueller glial cells after we bypass the hippo signaling pathway by a gain of function strategy where we use a particular form of JAP that is blind to the hippo pathway. And so when we finished this work, we were able to demonstrate that the cells that express the mutant gene, which is this group of cells, are, after we unrepress the signaling, these cells become highly proliferative. And so that doesn't occur in any mammalian species. So that was a big step. And then we also show that these cells can lose their glial cell identity. So these are the same group of cells. This is actually, I didn't explain this correctly, but this is actually single cell analysis that we did on those cells that we isolated. And we also found that these cells, this group of cells, not only lose their identity as glial cells and enter the cell cycle, but they start becoming a more proliferative more progenitor-like, so more like stem cells. And so after several bioinformatic analysis, we also found that indeed these Mueller glial cells require a particular metabolic niche for undergoing proliferation. And from there, I have some hypotheses that there is a particular niche, metabolic niche that is required for these cells to actually at some point become neurons. And so from all this work, I started to come up with my own ideas and trying to see how I can grow from there on my own with my own independent research. So just to summarize this part of the talk, basically there were 3 years of undergraduate research that introduced me to the retinal field, 5 years of graduate school that really tests my determination and perseverance. And for the next 3 years of postdoctoral training, what I'm doing is preparing to develop those new ideas that will allow me to be an independent researcher in retina. So that's my future plan. But the real question for this particular seminar is what were the most important components that allow me to advance in my science career? And I think that without doubt, I believe that being in the right environment matters. Being physically present at the university campus, and I think that under these circumstances that is more obvious, and we appreciate that more now than ever. And so I started my career science at UHD, and I like this cover from winter of 2011, which describes how the student population, how diverse it is. Many of us come from different backgrounds, but we all share a few things, and we all are curious. We all want to explore, and we all are looking for professional growth. Many students also are taking care of their families while attending school. They have full-time jobs. And in a personal note, when I was working 2 jobs while I was attending UHD throughout my first years. And then at some point I realized that to be able to graduate in a reasonable amount of time, I needed to attend school full-time. And that is where my goal became to join a Scholars Academy. And so I made a great effort to become part of it. And Scholars Academy at the time provided me with the financial support that I needed to spend a lot more time in campus. And so after that, I started to realize that faculty at UHG were very approachable, that they listen to you and listen for you, and that they have this remarkable ability of and willingness to share their knowledge. And I think that those are 3 major characteristics of mentors. So that was key for my to keep going forward. So Scholars Academy not only provide that financial support, but they provided numerous opportunities to meet faculty from other institutions, and also to attend local and national meetings and conferences. And that was key because I started to then start asking questions to what I'm going to do next, where I am going when I graduate from UHD. And this is essential because many of us are first generation college students, and we don't develop that instinct to see how our career paths look like. So we need that guidance. And so that was key. And I appreciate that overall. And the other benefit about being in the right environment is that you start developing these long-lasting professional relationships. And one thing that we have to learn is how to maintain them. And those relationships will be with your peers as well as your faculty mentors. And at some point you start defining, unconsciously you start defining what success means to you. And I think it's a very personal definition. What it means to me to be successful this semester, this year, once I graduate. And so all that is really important. The other major part is I do credit my interest in research to my undergraduate and graduate mentors. And so during my first experience, I worked in the chemistry lab of Dr. Byron Christmas at the time. And for first time, I experienced what it means to be in a lab. And here is where I experienced that, or I realized that it requires a lot of resilience, because you fail repeatedly, or your experiments might fail repeatedly, and you have to remain optimistic and focus on the big picture and keep going. And that's not always easy to do. But what I didn't know at the time is that that experience, that part was not going to change. Now, as a postdoctoral trainee, I go through the same things. There are a lot of experiments that don't work, and we have to keep persistent, and we have to really keep our motivation and optimism going. And so that part matters a lot, the first experiences in the first lab. And during my second undergraduate experience, I started to learn how to function independently in the laboratory. And so I started establishing that personal connection to the work that I was doing, and the sense of ownership. And that is where your mentor is a big part of it, because when they're really interested in your success, they ask the right questions. The questions that will make you go through the scientific process on your own, and the questions that make you see what is the next logical question for that particular experiment. And if you develop some level of independency when you do during your undergraduate research experience, then I'm pretty sure you're well prepared for graduate school. And so by the time I reach graduate school, the expectations are really, really high. You start being asked about the data, how many experiments you finish in a week. What are the abstracts, what are the publications, and all that is very good, and something that you do need to be able to integrate your ideas with the field that you work in. But more important, I think we have to try to balance and cultivate the relationships with the people that we work with. And so my one of the most important experiences is to be able to connect with people that look like me and that go through the same feelings. And that is important because you find a lot of support just by talking about it. When you start to combine all this, and if you find a mentor that takes the time to know you well, your self-confidence really goes up, and you grow. And I'm not gonna lie, there are a lot of graduate students that struggle because of the relationships with their PIs. And one thing that I think about nowadays is that when you move from your undergrad to graduate school, you should have an idea of what kind of a student you are. And so that should be a goal. That should be something that you should learn from undergraduate research. And that is because we at the end are life learners. We never stop being students. And so it's better to know from the beginning what kind of students we are. And so from there, responsibilities grow, and you have to start getting out of your comfort zone. And as you move on, you will learn and identify your own weaknesses, and you must reflect on your own about them and get out of your comfort zone in many, many occasions. And so during my postdoctoral experience, right now, I am trying to balance that collaborative and individual work, identifying what I am going to do as an independent researcher, and determining what kind of mentor I will be. And you learn from every single experience that you go through. But because we live in a complex society, and that's not a cliche, now as we move, we encounter a lot of social and cultural barriers. And one of the comments that I hear in some of these workshops, that the professional development workshops that I attend, is that many of us might end up in institutions and in departments where we are the only woman, the only female woman in the only minority and woman in the department. And so basically what that means is some of us will be in the front line. And to be honest, that's a very, very scary thing. But, you know, that will happen for many of us. So when that happens, there is a lot of support. And so 2 years ago, I met Dr. Elizabeth Zuninja Sanchez in one of the conferences that I attended. And she pointed out at me at the many resources that are actually out there for minorities in science. And so these are two of the major programs that I participate in. And they really provide a lot of support. You go to these workshops and you start talking to others that feel like you, that look like you, that are going through the same experiences. And now you brainstorm and you think about what kind of solutions or what kind of strategies you will use to overcome all those social and cultural barriers. And that is very powerful. And so in one of those workshops, and I want just to share this part, I learned about this diagram, which is in one of the books of Stephen Covey, in which you have 4 quadrants. And we can use these in a daily basis, in a weekly basis, in a monthly basis, in a yearly basis, where you have 4 quadrants, and you have to organize your life based on this. What is urgent? What is important? And so those things that need to be done right now. So if you're a student, what is that you need to pass that test right now, right? So what you need to do is study and pass that test. Those are the things that are urgent and important. Then you have quadrant number 2, where it could be the fun quadrant, where you find the things that are not urgent but are important. And this is when you did that internship, for example, and you did some work and you want to make sure that you're going to be in that publication for that work that you did. So you have to plan a lot. And if you develop the right habits and if you follow up these by planning, you actually make this the more fun quadrant. So it's the one that has the most fun. Then you have, as you move on in your professional career, you start encountering what is urgent but is not important, or could be not as important. And so that's because you are going to become part of an institution. So you are going to have to be part of meetings and things that you might not consider as important, but they are urgent and they are necessary. And I haven't reached this point yet, but I hear that from a lot of the things from the workshops that I attend. And so because we are only humans, we have all those things that are not urgent and not important that we must eliminate, that we have to change certain habits. And so that is part of this, and writing it down, it helps a lot. So this diagram, I have found it very, very powerful. And what I wanted to leave you with is this diagram that is also part of one of the workshops that I attended last year. And it's about how we start thinking about becoming leaders. And I think that the major strategy to get there is to fill up this particular diagram, in which you will have to identify what kind of support you need, and we have to identify who can actually fill each box and who can participate in that part of our lives. And this is a multi-mentorship map. This is basically allowing you to find the right resources and the right people for the right support. And so this is one of the things that I I plan to implement in my mentorship philosophy once I get my faculty position, in which I'm going to encourage the young to think about this in the earliest stages, because I think that if I had started working on this multi mentorship map early, I would have struggled a little bit less in certain areas. And so the support is there. And so that's what I wanted to the message that I wanted to give you today. And I have a lot of people to thank and a lot of institutions. And then I want to emphasize Scholars Academy.
I think you froze, Elda. Elda, I think you froze up. Can you hear me, Elda? Ah, okay.
Did
Oh
it?
yeah, you froze. You right at the end, you froze.
Oh no! In which part?
But that's all right. Well, the very last slide, actually. After Scholars Academy, we didn't hear anything else.
After.
Go back to your last slide. It was the last slide. The people you were thanking. I think we got most of that.
Did all this went through? Because this is the most important part.
The little flow chart, we got that. It was your last screen.
This one?
Let me see. No, we're not seeing it. Share your screen.
I am.
Let's.
Oh, wait.
Oh,
mm, okay, let me see.
it says you start. There we go. Yeah, go to your last slide, I believe it was. Then we'll get a few questions.
Okay, so this one?
No, you're on that's the first one. Go to the last. All the way. Right. Oh yeah, this one. That's where we were.
I think that
Yes,
I think that is lagging. I think is the communication is lagging. So I am already on the last slide.
Okay, yes. There, I see it. So you were just thanking everybody.
Yes. Oh, so I have a long list of people to thank, and institutions and programs. I didn't say much on this one, so just I just move
Yeah.
on into the questions.
Okay, good. All right. Do we have any questions? I see some of, we have two people that are present that are actually already admitted. One to UT Graduate School of Biomedical Sciences, to Kesey. And
Yes, I
then
know her.
Linda Motamed, she's going to pharmacy
Mohammed,
school.
yes.
No, optometry. What is it?
No, I'm going to pharmacy school. Hello, everybody.
Do you all have any
Hi.
questions?
Yes, I'm going to Texas Southern University. Yes, pharmacy school.
Pharmacy. Okay. Well, I had a question for Elda. I think wonderful presentation. Thank you so much. And I do remember, because I had just come and you were, I think, a sophomore or junior. And I remember when you began working with Dr. Johnson just from afar, and you just came out of yourself. You know, you were so quiet and shy, and the research really helped you see yourself with more confidence. But tell me, based on that, here's the question. So in an honest but frank way, you know, are there uh, real barriers for women and for minority women as they move? I mean, I heard you say that, but how, you know, did you encounter them early on? Were you able to deal with? Obviously you were, but anyway, so were there barriers for women and minority women, even more so, as you went through to your successful piece right now?
Yeah, so I would say that yes, although, so I didn't talk about this, but I am an immigrant. I am a Latino. I'm from Colombia. And in my culture and in the community that I grow up, women are always underrepresented in every single way and underestimated. And so I think that I came with that. I came with the stigma that, you know, um, the belief that the only way for to see you as a successful woman is to have a family and take care of your family, and that was your main role. And in a way, inside of me, I feel that I was fighting that and say, no, we have to do more. But you're right, I was very, very shy, and I wasn't the person that was going to scream it aloud and say anything. And so I think that what I did is I focused on the job. I focused on the research a lot. And you were right. You were exactly right. Once I started to participate in research, because of the advice from many faculty in the natural science department, I was able to come out of the shell and become a little bit more confident. I think I'm still working on that part. But one of the major barriers for me right now is, for example, I'm working in a highly collaborative environment in which we have to go and talk to that famous PI. And so a lot of the people is still in my field, and a lot of the departments are still dominated by males. And so naturally, I am afraid of going there and asking for that attention, because I feel that I'm asking for attention, and I feel in a way that I'm being needed. And that's completely ridiculous. We have to overcome that and start thinking that you will speak to that person as, you know, as if he was the person you work in the lab with. You cannot see yourself less than that person. And I think that is very difficult at the personal level. And I am sure, because I go now to this workshop, for years I thought it was myself. For years I thought I was, no, it's me being, you know, this person that never did, that you know, came from a Latino culture where that is the belief. And once I go there, you validate those feelings. Once I start joining these workshops, I validated that, because many people feel exactly the same way, and they go through the same experiences. So what you do is, just by going and saying and sharing, you come back to your job with a lot more strength. And with that, that's when you go now to talk to that PI that you were afraid to talk to. And, you know, as a postdoc, I'm not talking about when I was a student. This is happening to me when I'm a postdoc, and after all these years. And so it is something that we have to work on. And I think there is a great effort that these programs sponsored by the National Science Foundation and NIH, they're doing a great job. They're actually bringing us together, and it's going to take some time, but it has to be done, and we have to do it. And so that's the major thing in a personal level. And so the support is there. You are going to be able to grow as a scientist, because there is also a lot of money in STEAM science right now. But it's not just that. It's about the personal part, you know, and so that's why I appreciate from those programs.
Wonderful.
I hope, okay.
No, that got it. That got it. Linda, do you have anything?
No, I just watching and learn. You really have a good, excellent presentation. I always scared about this part of, do I have to practice that?
You do. You need to be able to, you
Yeah.
know. Well, she's had years of practice.
Yeah,
Yes.
I have to. Yeah, because always I always scared somebody ask question and I don't know and I cannot answer, and or maybe my language doesn't let me to understand, or I lose my confidence at this part.
Yes. And the language is another part. So I moved here in my early 20s to the US, and I did not speak English. I learned English at UHC. There used to be a program. I don't know if they brought it back. There used to be an English learning program. That's what I went. That's when I first time I went to UHC.
Yeah, I did the same way. Yeah.
And so what I can tell you is the more you do it, the more you practice, the better you get at it. Yes, we all feel the same fear. And even after so much practice, I still feel that, and still feel, wow, I might not understand the question, but that goes away. I promise you that goes away. And even if you don't know the answer, you find a way to say, well, I don't know that, but this is how I would find out the answer, or somehow you get more confidence with time. You just have to be there and allow yourself, expose yourself a lot.
I can tell you something funny. I went to Walmart and I wanted to talk to somebody, but I at the moment I forgot things I wanted to tell. And I have a 5 years old son, and he was watching me, and he saw I'm in frustrating time. I cannot transfer my feeling, and he told me, "Mommy, you want to tell this?" And I told, yeah, I want to. He reminded me the vocabulary. I lost that.
Yeah.
Really, yeah, it happened
Well,
already.
I wanted to share, it's not a question so much, Elda, but very good for me to be reminded because of how important the national conferences are. You know, we struggle with being able to afford sending students, and yet hearing, and I do know that they're important, but hearing you say, you know, that this really can impact your network and open the possibility for connections to the next level is so important. And I appreciate you putting that in there. You know, sometimes you get too close, you worry about the money, but you forget the benefit.
Yeah.
Right.
Yes.
So even send you versus everybody is good.
Exactly. And that's
Well,
that's something
anybody else have a question? Great. Well, I think, and we are at 45 minutes, so we don't want to take your time away. You're very busy and you got finals to grade. Thank
I
you
do.
so much for being a part of this. And